Our topic today is the development of antibody-drug conjugates with emphasis on safety and ADME consideration for development of this molecule. Let's consider it evidence that biotech is interested in obtaining business for development of novel modalities, including biologics, such as ADCs. We'll consider or present introduction to antibody-drug conjugate. We'll talk about the mechanism of action of ADCs that are approved by FDA, and we'll also talk about drug interaction potential of these modalities. And later we'll go into identification of drug interaction potential of ADC payloads, and you'll see throughout the presentation why we're focusing on properties of the payload from the perspective of DDI. As a matter of introduction, antibody-drug conjugates are a relatively novel modality which is designed to deliver highly cytotoxic agent to the selected cancer cells using a guiding mechanism, and monoclonal antibody serves as this guiding mechanism. Precision of the delivery of this toxic molecule, also known as a payload, is based on a specificity of antibody and the expression of a targeted antigen. Therefore, antigens that are expressed by disease state, preferably exclusively, but not healthy cells, are targeted. This is ideal situation. We'll see that this is not always possible. There are four components of ADCs are, of course, monoclonal antibodies that direct payload to its target cell. Payload itself is a cytotoxic molecule providing therapeutic effect, and linker is a molecule that is responsible for stably attaching payload to the monoclonal antibody in the way that does not interfere with antibody recognition of its antigen, but it also allows to release of intact payload in lysosomes when the rest of the conjugate undergo hydrolytic degradation. For your convenience as a reference, I prepared a table listing antibody-drug conjugates currently in the clinic, and on the further slides we'll provide certain comments about indications for which these drugs were approved, what they are antigens or targets. Therefore, we will learn what are the mechanisms of the individual ADCs, and also list information about box warnings, since we are concerned about safety and side effects of these molecules. So, in the United States, there are 12 ADCs currently on the market. There was one more approved by FDA, but it was withdrawn after 2 years in the market due to the lack of efficacy, and it was ADC against multiple myeloma. Interestingly, it was carrying a payload of auristatin F, and the rest of the Deutens or ADCs containing auristatin using auristatin E. There are also two more ADCs, one approved in Japan and one approved in China, but we will not talk about them. They are not approved in the United States. So we'll go through some comments about the indication, targets, and payloads. On indication, I would like to say that hematological malignancies such as leukemia, Hodgkin's lymphoma, anaplastic large cell lymphoma dominate and are followed by solid tumors, HER2 positive breast cancer, urothelial, cervical and ovarian cancers, non-small cell lung carcinoma, as well as HER2-positive esophageal junction adenocarcinoma. So from this, we clearly see that ADCs currently approved are only for oncology. Targets are epitopes expressed uniquely on the cancer cells. Jointly, they are considered to be a cancer surfaceome, which currently enumerates about 30 molecules that are considered uniquely expressed on the surface of the tumor cells and also are considered druggable. This is a summary provided by Disco Pharmaceuticals. Selective markers are targeting by multiple ADCs. For example, HER2 antigen is targeted by trastuzumab emtansine, carrying microtubule inhibitor, and trastuzumab deruxtecan, carrying topoisomerase inhibitor. Similarly, antigen CD22 is addressed or treated, if you will, by one ADC carrying a DNA cleavage agent and another carrying apoptosis-inducing agent. First of these examples, that is trastuzumab, is an example of a case that successful antibody can be conjugated with multiple or different payloads for treatment of a given disease, allowing, in a sense, a two-pronged attack on a given tumor cell type. Going to the payloads, all currently utilized payloads are derivatives of natural products, although they are too toxic to be administered directly to the patients without guiding mechanisms. Therefore, they have to be administered with some mechanism that will reduce exposure to the normal cells. Further on the therapeutic payloads, we can organize them according to their mode of action. They are DNA cleavage and cross-linking agents, microtubule inhibitors, topoisomerase inhibitors, inducers of apoptosis, and there are also novel payloads being developed for oncology, including DNA alkylators, RNA polymerase inhibitors, as well as agents promoting or amplifying anti-tumor response. And this is exemplified by the payload that is a toll-like receptor 8 antagonist. For non-oncology, because this modality is going to be utilized outside of this field, other therapeutics or payloads will be used. And in many cases, they will be less toxic, and they may be already established and tested agents. Going further to understand why and where toxicity of ADCs may happen, we'll go briefly into a schematic representation of mode of action of this modality. On this diagram, we have ADC molecule, in this case equipped with a 4 molecule of payloads. Through the interaction with the antigens present on the surface of the tumor cells, the construct or conjugates enter the cell and is processed through endosomal lysosomal pathway, undergoes hydrolytic degradation in the lysosomes. Payload is released into cytoplasm and finds its way to its molecular targets, such as DNA or microtubules. Interaction with both elements may lead to apoptosis and death of the target cell. This information presented in this format gives us the impression that ADCs are specific, therefore safe and effective, but really the essence of our presentation is to focus on a toxicity and adverse effects of these molecules. And to understand where and why these toxicities were observed, we can take a look at a more detailed look of mechanism of action of these drugs. Again, here we have ADC, which is present outside of the cell and can enter the cell through targeted antigen, but also can enter the cell that is either normal or tumor cell through interaction with other receptors, such as receptors against a constant fragment of antibody or other receptors structurally related to targeted antigen. And C-type leucine receptor is one of the examples of such a receptor that would be responsible for entrance of ADC into the normal cells. When ADCs are outside of the cell, they can enter the cell through other mechanisms, such as non-specific endocytosis, and also unconjugated toxic payloads can enter the cell through either passive diffusion, transporter-mediated process, or micropinocytosis. Similarly to previous diagram, we see transfer of the ADCs from endosomes to lysosomes. In lysosomes, we see hydrolytic degradation, release of the payload, and then a distribution of the payload to its targets. But it's also noticed that some of the payload released from the lysosomes can escape cancer cell even before this cell is being killed, either by transporter-mediated mechanisms or by diffusion. In these cases, toxic molecule of the payload will interact or otherwise kill normal cells present in the vicinity of the tumor, and this is called a bystander killing effect. Therefore, we see that a lack of total specificity between antibody and antigen is one of the means for toxicity. Non-specific entry through other mechanisms is another. Uptake of toxic payloads separately from ADC is another. And for that phenomenon, that is conjugation here, linkers are responsible because not all of them are as stable as we wish they be, and almost in any case, some of the ADCs are releasing their payload prematurely in the plasma. Associated toxicity problems are demonstrated by presence of box warnings. This is essentially a reference slide for you. I would only say here that box warnings were assigned to about 75% of approved ADCs, which is a recognition of serious toxicities associated with these drugs. But considering complexity of ADC, these three major elements, properties of the payload, that is high toxicity of these molecules, monoclonal antibodies present, and linkers, as well as disease processes, we recognize that all of these factors may contribute to observed toxicities. But for example, I would say that out of 4 auristatin-containing antibody drug conjugates, and auristatin is the most commonly used payload, only 2 of them has box warnings. That means that auristatin by itself is not necessarily cause of toxicities that would call for issue of box warnings. We'll go briefly into safety aspects of different elements of ADCs. Epitopes expressed uniquely on the cancer cells, as I indicated earlier, are ideal to reduce off-target toxicity of these molecules. But safety consideration related to that is that ADCs, as I indicated on a previous diagram, can bind and enter normal cells as well. Antibodies with rapid internalization upon binding are preferred because molecules that are binding to the antigen but not internalized can be a subject of hydrolysis and premature release of toxic payloads. Effector functions that are associated with antibodies, that is with a constant portion of the antibodies, can also create a safety issue for normal cells. For targeted cells, they can be a part of a therapeutic effects, but for healthy cells, antibody-dependent cell-mediated toxicity is a serious toxicity issue. One of the ways that people think about improving on this is utilizing bispecific antibodies that could base their entrance to the cell not by interaction with one protein on the surface of the cell, but two of them, therefore greatly increasing specificity of the interaction. But bispecific ADCs are at the moment not a part of approved repertoire. Further on payloads, I would say that the safety consideration is that drug transporter substrate potential exists for payloads, and as I indicated on one of the previous graphs, that contribute to cell bystander cell killing in tumor microenvironment, killing of abnormal cells. Also, lipophilicity supporting passive diffusion can enhance off-target toxicity. When payload is released in the plasma and it's highly lipophilic, it can enter just due to that property normal cells. Linkers technology contribute to stability of excuse me, ADCs in plasma and certainly contribute to uniformity of the conjugates, that is drug-antibody ratios. Both are two important determinants of ADC safety. But catabolism of linker in plasma and in human lysosome is also of concern. Linkers need to be designed in a way that are very stable in plasma and prevent premature release of cytotoxic payloads there. But also in the human lysosomes, they need to be completely hydrolyzed to the smallest possible molecules that will not create any potential issues related to further interactions with these molecules. Our lysosomes contain a range of hydrolytic enzymes. Among them are catepsin proteases, which are serine catepsins A and G. These are responsible for cleavaging of linker used by auristatin, for example. Others are aspartic catepsins like D and E, and cysteine catepsins, it's a group of 11 hydrolytic enzymes. Other are, among others, we find acid lipase, sulfatases, and nucleases, and other enzymes organized in the lysosomes to degrade any naturally occurring cell debris. Following this introduction, we'll go briefly through a couple examples of different toxicities observed for ADCs. On this one here, let's say that 3 different antigens on the top of urothelial carcinoma cells, TROP2, Nectin-4, and HER2, are addressed by 3 different ADCs. All of them are highly effective for intratumoral drug concentration and effective killing of the cancer cells. Therefore, they have an ability to lower systemic distribution and off-target toxicity. First, we'll focus on sacituzumab govitecan, which utilizes TROP2 as a mechanism of entry, and it's equipped with 4 molecules of SN38, which is a metabolite of irinotecan. Main toxicities associated with this ADC are diarrhea, alopecia, fatigue, and cytopenia. Please note that diarrhea is a side effect for which box warning was issued, and it's a toxicity that differentiates this particular ADC from other ADCs utilizing different mechanisms of entry for the same urothelial carcinoma cells. Therefore, we can look in more detail about the mechanism of action of irinotecan, or more specifically, its metabolite SN38, which is a payload of the antibody in question. So, sacituzumab govitecan is an antibody drug conjugate of SN38, approved for breast and urothelial metastatic cancer with a warning, and warning is related to genetic condition in which individuals carry allele 28, allele 28 of UGT1A1, which is associated with the lack of enzyme activity. Hepatic UGT1A1 and UGT1A9 inactivate irinotecan, and patients with allele STAR28, especially homozygote, are at higher risk for neutropenia, anemia, and diarrhea. Therefore, FDA recommendation is to avoid concomitant use of this particular antibody with UGT1A1 inhibitors or inducers. In case of application of SN38 using antibody drug conjugate, we are not concerned about the activation of the irinotecan, which is done by carboxylic esterases, but we are concerned about the availability of enzymes that deactivate that molecule, and in the liver there are more than one UGTs capable of deactivating SN38, but the question is different or situation is different in intestine, and in the case of polymorphism leading to lack of activity of UGT1A1, SN38 is not neutralized, therefore diarrhea is a major toxicity associated with this ADC. Going back to other ADCs utilized for urothelial carcinoma, we note that they are very similar, except mechanism of entry they utilize. One is utilizing antigen of Nectin-4 and one is of HER2. Otherwise they are similar. Both carry auristatin E, microtubule inhibitor. They have a similar drug to antibody ratio, around 4. Both of them are using the same type of linker, and they are also the same class of IgG, specifically IgG1. There is a differential toxicity between these two antibodies, which is skin toxicity observed with enfortumab vedotin. Therefore, since we indicated that mechanism of entry of this ADCs differ by the antigen they use, we'll look at the expression of Nectin-4. And on this diagram, we see that Nectin-4 is highly expressed in skin, and expression in skin is about at least 4 times as high as in the bladder, and we're making comparison to the bladder since we're talking about urothelial carcinoma. So we can reasonably speculate that skin toxicity observed for enfortumab vedotin, but not disitumab vedotin in urothelial carcinoma, are related to mechanism of entry or selected antigen used to develop these ADCs. Further, we'll give one example of metabolism-mediated interactions. We'll focus on an example of brentuximab vedotin. Vedotin means carrying auristatin E as a payload. This ADC was approved in 2011, and at that time this was already recognized that drug-drug interactions involving antibodies are typically limited, but the cytotoxic portion of the conjugate can be subject of metabolism-based drug-drug interaction. It was demonstrated that auristatin E is a substrate of CYP3A and PGP, pointing out to the victim or object potential for that molecule. Auristatin E is also an inhibitor of CYP3A4, but not other CYPs. In clinical study, brentuximab vedotin did not affect pharmacokinetic of midazolam, which is a sensitive CYP3A4 substrate, but rifampin, an inducer of CYP3A4, decreased plasma exposure of auristatin. Ketoconazole, CYP3A4 inhibitor, increased plasma exposure of auristatin. And I would like to say that at least a portion of auristatin that was measured in the plasma in this study first entered tumor cells or the hepatocytes, where it was either metabolized or left the cells intact by either passive diffusion or some cell or drug transporter mediated efflux. From the drug label, we therefore read that concomitant use of strong CYP3A4 inhibitors or inducers or PGP inhibitors has the potential to affect the exposure to monomethyl auristatin E. Our presentation would not be complete if we would not go into essence of regulatory documents. And we'll focus on regulatory guidance for ADCs, which is provided by International Committee for Harmonization M12 guidance, soon to be approved, we hope. This regulatory guidance makes two significant points for ADCs. First one is that for ADCs, the small molecule drug component, which is conjugated to the antibody component, can be released in unconjugated form, and this is related to the limited stability of the linker or a construct. Therefore, the drug-drug interaction potential of both the antibody and the small molecule drug component should be considered. In many cases, however, it's recognized by FDA that the systemic concentration of free drug may be too low to act as a perpetrator in vivo, and it was demonstrated by the case of antibody we discussed, or antibody drug conjugates we discussed before. As far as the DDI potential of antibody itself, it is well recognized that antibody undergoes complete hydrolysis in lysosomal compartment and practically does not carry potential for drug-drug interaction. Second point made by the guidance in relation to these molecules is that it's important to understand the formation, distribution, and elimination kinetics of the small molecule component and to assess the systemic exposure of the small molecule drug component of ADC. And in some cases, guidance says it may be necessary to evaluate small molecule component as a victim drug if increased level of a free drug may be associated with a safety or toxicity concerns. Based on these regulatory guidance, we have a certain strategy for evaluating or characterization of ADME properties for ADC payloads. We discuss with our clients or suggest identification of major metabolites of ADCs in plasma, lysosomes, hepatic or tumor subcellular fractions. We also recommend evaluation of major metabolites as a victim and as a perpetrator of drug-drug interaction when appropriate. And that would follow with the evaluation of the data according to the guidance recommendations. And we are, of course, eagerly awaiting to see final version of these recommendations. For practical perspective, these recommendations are translated into certain studies. We're looking into metabolism-mediated interaction to answer the question, is the payload a substrate of drug metabolizing enzymes? And this question would be answered by conducting metabolic stability studies and reaction phenotyping studies in appropriate test systems, such as hepatocytes, microsomes, or recombinant enzymes. Further, we ask, is the payload an inhibitor of drug metabolizing enzymes? And we conduct evaluation of direct and metabolism-dependent inhibition of CYPs and potentially UGTs in microsomes and hepatocytes. Is the payload an inducer of drug metabolizing enzymes? And again, we conduct a standard induction study in cultured hepatocytes. We also looked at transporter-mediated interaction potential. Is the payload a substrate of drug transporters? And in cell lines, membrane vesicles, we conduct drug transporter substrate potential studies. So same way as we do for small molecules. Is the payload an inhibitor of drug transporters? Again, in cell line or membrane vesicles, we conduct drug transporter inhibition potential studies. In some cases, we have a capacity to evaluate metabolite-mediated interaction, and questions would be analogous to those asked before for a parent molecule, if you will, and therefore multiple test systems would be applicable. Last but not least, I would like to say that our disease state business unit can provide materials for characterization of differential expression of tumor antigens. And these materials would be slides, paraffin blocks, flash frozen tissues, or even isolated tumor cells. And the same tissues or cells can provide a test system for selecting most specific antibodies to be used as a delivery mechanisms in ADCs under development. In conclusion, I would like to say that antibody-drug conjugates are already a very promising modality addressing needs of the patients which are in a very dire circumstances. I must note that these modalities currently are recommended for patients that did not receive relief of the disease from at least one or two previous rounds of approved treatments. Very often these patients are elderly and generally described as fragile, which certainly can contribute to some unfortunate events that has been observed in clinical trials of ADCs. I'll also say that highly cytotoxic payloads utilized in ADC in oncology present their own challenges for development of safe and effective molecules. As I indicated earlier, they are too toxic to be applied without a directing mechanism. And if they, by one mechanism or another, are separated from that direct or directing mechanism, they present a serious toxicity issue. Multiple mechanisms involving individual components of the conjugates, disease state, and patient's characteristic are all likely to contribute to observed toxicity of antibody-drug conjugates. And lastly, I'd like to indicate that some of the critical properties of ADCs related to their toxicity can be improved based on in vitro studies. And I'd like to thank you for your attention.